Thanks for inviting me. I'm delighted to be back at Carnegie Mellon. So I'm going to try to defend to you why is this crazy idea of trying to map the activity of every spike from every neuron. I wouldn't say in the entire human brain, that wasn't actually our proposal, but more of a complete neural circuit, either in an animal or a human, and why this is a worthwhile enterprise. And actually, after Steve and Sean, I hope that you're at least convinced how this type of technological pushes could tremendously help already in terms of brain-computer interface or prosthesis. But a fundamental argument has to do with something even more basic. So I'm passionate about understanding how the cerebral cortex works. As you know, it's the largest part of our brain. It covers our encephalon. And it's essentially what makes us humans. We know from human patient studies and from animal lesions that all cognitive abilities are essentially buried somewhere in there. And the site of all the mental diseases, many neurological diseases. And I think it's fair to say that we don't understand how it works. We don't have a general theory that can explain how the cerebral cortex gives rise to our minds. And this is of utmost importance for mankind because we define ourselves by our minds. We are actually an intellectual species. So the minute that we can understand how this works, we'll understand ourselves from the inside for the first time. So it's going to be a turning point in the history of mankind when one finally understands the function of the cerebral cortex. So this has been realized already more than 100 years ago, and there's been generation after generation of researchers throughout the world building on the work of each other to try to tease apart these circuits. The cerebral cortex is built out of these laminar circuits stretched for about 2 millimeters from the top under the skull, the pupil surface, to the bottom, down by the white matter, and is composed of this mixture of excitatory and inhibitory neurons connected in very complicated fashion, or at least in ways in which we don't understand. And somehow out of the firing of these cells, our mind emerges. But there's no magic. There's nothing else in there. I mean, it's essentially just a bunch of neurons firing. So we can actually decode the firing. We may be able to actually understand how our mental states occur and what are the physical nature of our mental states. The central hypothesis for 100 years is that we're dealing with some sort of modular system, that evolution has developed the cerebral cortex very late. It's something quite recent, and it's grown tremendously, which has the typical hallmarks of a gene duplication or a developmental duplication type of strategy. And according to this hypothesis, we could be dealing with a simple unit, which some people have called columns or chains that would go vertically. And the human cortex would have maybe 1 million of these units. We don't know how many, but maybe the mouse would have 1,000 or 100 of these units. And the reason why humans are intellectually more able than mice is just because we have more of these units. So from that point of view, if you could understand one of these units, even in a lowly mouse, you could actually decode the function of these impenetrable jungles. And if you could do that, also you have to realize that different parts of the cortex are involved in completely different computations that have apparently nothing in common. So we could be dealing with some sort of biological Turing machine, with some sort of strategy for computing that could be used to compute any problem that can be computed. So that's the reason I refer this problem of understanding the unit of the cerebral cortex as the double helix of the brain. It could be, it has to be something simple because it has to be the common denominator from all these different computations. It cannot be something very complicated because otherwise, just by nature, you won't be able to do vision and motor control at the same time. So we're hunting, and the whole field, for 100 years for this double helix of the brain. We hope that once that piece goes in, then someone could actually build together, put the architecture of a general theory that could explain the mind. So why hasn't it happened? What people have been doing for 100 years is been studying individual neurons, and this starts with the work of traditional anatomists like Raymond Cajal, who used the Golgi method to label single cells and draw their exquisite morphology, the drain dextrins and axons, and catalog the cell types across the brain. And this gives rise to the idea of the neuronal doctrine, that the unit of function of the nervous system, of function and structure, is the single neuron. And because of that, the effort should be focused on understanding what single neurons do. And this anatomical neuronal doctrine evolves into a physiological neural doctrine with the development of the tungsten electrode. In this case, this is actually a homage to David Hubel, who just died a few days ago, and he was the person that first pioneered this tungsten electrode for single unit recording. And the idea is that the single neuron is the unit of the function. You characterize the electric field, and in some cases it could be very complicated. Horace Barlow argues that there are grandmother cells, that we have neurons in our brains that can actually detect our feature detectors for our grandmothers. So the physiological neural doctrine goes hand in hand with the anatomical neural doctrine, okay? But I want you to go through the following exercise, a thought experiment. Imagine you're a UFO scientist and you land on planet Earth and you land in New York and you see the Empire State Building, okay? And you're a very good scientist, very well trained, and you say, okay, you know, I'm going to understand how this bloody thing works. And I'm going to do that by taking it apart. And you figure out that it's built out of atoms. And you start studying the atomic structure of the Empire State Building. So you can study it for many, many, many hundreds of years and will never figure out what the Empire State Building does. And the reason for this is because the function of a building, although of course it has to do with the atoms that build the bricks, it's actually written at the level of the architectural floor plans and the way in which they interact with people. And this is the pitch for emergent properties. Emergent properties are properties that happen in complex systems of many interacting units or particles from the interaction between the particles. And by definition, they're not present in individual particles. They've been studied by chemists and physicists for hundreds of years. This is nothing new. And it actually, our world is filled with emergent properties, starting with the solidity of this table. That's an emergent property of the atoms. You cannot understand really the solidity just if you look at the single atoms here. So just now go back to the brain, and any piece of the brain, wherever you look, you find that it's built out of hundreds of thousands of millions of neurons. This is not the cerebral cortex of the mouse, which is the preparation that we normally study. The PL surface is here, the bottom, the white matter, and every one of these white dots is a neuron. You don't see the dendrites or the axon. This is just the cell bodies. No? So just imagine trying to understand how this works by recording from one neuron in one animal, okay, and then you compare that data and go to another neuron from another animal, and go on and go on and go on. So this is just like trying to watch a movie at night in your TV if you can only see a single pixel. So I would argue that it's very likely that the reason why we don't understand how the cortex works is because we're dealing with a classical emergent system. This is built to carry out emergent level computations. And that the functional unit is not the neuron, it's the neuronal ensembles, the groups of neurons. And we don't know how many these ensembles, how many neurons do they involve, but for many reasons that I can argue with you later, I'm convinced that this is some sort of unit of functioning here. So from that point of view, what we've been doing in the past, using single neuron anatomy, single neuron electrophysiology, has been very fruitful, but not sufficient. So how do we tackle this problem? Well, you can imagine putting electrodes, okay? And that you could record from some neurons, but you're not going to be able to really get at the entire network, no? Particularly in very dense areas such as the cerebral cortex or most part of the forebrain. So with many other people, we've been arguing that the natural way to do this is with light. And this is actually also to highlight the pioneer work of Fred Lanning sitting in the audience and his culprit with me of pushing for the approach of using light to decode the function of these neural ensembles. And you could argue that imaging would be a field that could be defined as, from this point of view, as a microscopy of living cells, living systems. And it's an ideal platform. It's very interdisciplinary, which also makes Carnegie Mellon such a special place, because this is one of the few places where chemistry, physicists, biologists, engineers, and medicine is interacting together. So this is the type of techniques that have to be built not by individual fields, but by interaction between fields. And by definition, it's not just a single lab, but interaction between many groups of labs. Now let's go back to the cortex. Okay, let's use light to try to figure out how these modules of the cortex work. Well, you have to solve one big problem, and it's the fact that there are many different types of neurons, as Nathan was mentioning earlier. These are not an infinite number of types. We don't know how many, but there are definitely maybe dozens or hundreds of cell types, but they're all mixed on top of each other. So these are examples of some of these different types of neurons from the cat V1 or the mouse V1, drawn side by side so you can appreciate how different they are, but in reality they're actually physically on top of each other. So if you're going with light, if you're going with visible light, then you're going to scatter, because the brain has the sort of scattering properties of a glass of milk, so you're going to scatter through there and you won't have single cell resolution. And it's essential to have single cell resolution, because it's not the same if you record from a neuron that looks like this or a neuron that looks like that, because they're likely doing different things. So the idea is to use nonlinear microscopy to photon excitation, which through the nonlinearity that occurs at a focal point, that enables you to actually, first of all, have, well, through the fact that you use infrared light, you can actually penetrate deep into highly scattered media, and second of all, the nonlinearity gives you the ability to do optical sectioning in the middle of deep brain tissue. So my proposal is to use 2-photon as a general platform to decode these neural circuits in the cerebral cortex or in other parts of the brain, transform them into engineered physical systems that we can understand and fix when they're broken, and try to tackle this issue of the neural ensembles, assuming that we have to deal with an emergent level system. So to do this, you need tools to be able to image the activity of the circuit, and just like you like to watch the TV in your living room watching every pixel, I would say that it would be good to catch every spike from every neuron in a neural circuit. You need tools to be able to turn on the cells, because you need not just to watch, but to be able to manipulate or to turn off the cells, and finally to be able to play the piano. And by this I mean to arbitrarily turn on or off any neuron in any spatial temporal pattern that you want, because if you can do that, you can actually extract the transfer function of these types of electronic circuits, just like an electrical engineer would reverse engineer an electronic circuit by using a particular protocols to do that. So I'm going to give you a little bit of an overview of different types of technology that we've worked on that can be used to do this job. And I apologize if it's going to be a little bit superficial discussion of this technology, but I'll be around today, so you're welcome to ask me more technical questions if we run out of time. Let's talk about imaging, and just to illustrate how important this interdisciplinarity is, the best thing we have as a field for imaging neural activity comes from the work of organic chemists who design indicators for calcium that have very high affinity, and they change in the spectroscopic properties when they bind calcium ions. And this was very interesting from the point of view of monitoring calcium for cell biological purposes, until we discovered by chance that you can actually use calcium to indirectly monitor action potentials. The reason being that every neuron has calcium channels in its membrane, and when the neuron fires, these calcium channels open, and they're letting enough calcium to significantly cause a change in intracellular free calcium concentration. And this is illustrated here, where we're measuring simultaneously calcium using just an optical measurement, fluorescence, versus time. This is intracellular free calcium in the center of a neuron, and these are the pattern of action potentials that we're inducing into the neuron with an external electrode. And as you can see, every time the neuron fires, there's a reproducible increase in intracellular free calcium, which means that you can actually use this calcium to decode when the neuron fires. And in this case, the spike didn't occur, and there wasn't any blip in the calcium change. So you can use calcium imaging because you can load these calcium indicators en masse on populations of neurons. And this is an example of 500 neurons in a primary visual cortex, 10 minutes in the life of this circuit. Every neuron in the area is illustrated in gray, and whenever it's active, it's turning red. And this is by using calcium to monitor when the neuron is firing. So this gives you at least you start to see, I wouldn't say the entire TV screen, but a significant piece of it. In this case, it's 500 cells out of a population of about 180,000 neurons in the primary visual cortex. You can do this in living animals, and this you may have seen this type of movies before. but you can do it in awake-behaving animals, like this mouse here that is sitting under the two-photon microscope or watching a screen, and in the screen we're showing him the typical human and weasel type of stimulus to drive his primary visual cortex, and simultaneously we're measuring calcium in a particular layer, layer 2-3, and you can see in red how the neurons are never firing together, they're always firing as part of a group. So this is what I mean by neuronal ensembles, that we're dealing with essentially coordinated groups of many neurons. Now, calcium imaging can be used up to a particular frequency of firing, but it's not really what we need to really measure to follow the electrical activity of the nervous system. We need to measure voltage, because this is actually what the nervous system is using as the coinage of its information transfer. And measuring voltage optically is very complicated. It's a very tough problem, and it has to do with the very small dimensions of the plasma membrane, about 4 nanometers, and the fact that the electric field extends only for probably about a nanometer or so beyond the plasma membrane. So this is the Debye length of the electric field, which means that if you want to measure this optically, you have to put your chromophore within this little tiny piece of space. And this is technically very challenging because it's a very, very small area of the cell. And second of all, it's a very sensitive area because any photodamage you can cause here breaks the membrane and kills the cell. So this is a problem that I would argue, though we've worked on this ourselves, is not been technically solved. We still probably a few orders of magnitude in terms of signal to noise where we want to be with voltage imaging, so that we can actually replace calcium imaging and turn on to image voltage instead of calcium. We've done a little bit of voltage imaging development ourselves. We've used a nonlinear optical effect called second harmonic generation, which is hyperscattering, which happens to be electric field dependent. It linearly changes with electric field. When you change the electric field in a neuron, here you can actually change the second harmonic generation that it emits, and you can use chromophores such as transretinal to label the cells and see the plasma membrane and use that to measure voltage. Second harmonic is, by being a nonlinear phenomenon, can be used also to do optical sectioning, and it uses also the same lasers that I use for two photons, so you can also penetrate deep into the brain. This is an example of measurements of second harmonic. Another approach is to use genetically encoded voltage indicators that are based on green fluorescent protein or their derivatives, and you have to stick them next to the membrane within the Debye length. And this is an example of one which is called ArcLight, developed by Vince Pierboni at Yale. And in this case, we're just measuring voltage in these dendritic spines in a pyramidal neuron in a brain slice using two-photon excitation, and you can see when the neuron fires action potential, you can see a change in voltage in these dendritic spines, but notice also the very poor signal to noise I was mentioning earlier, in which you can see the change, but the red brackets here are sort of the level of noise, of confidence of our noise. No? And just to make a pitch for how important interdisciplinary science is, let me bring you another idea. This comes from the work of people in the field of quantum optics, and they've worked with nanodiamonds, which are the imperfections in diamond lattices that instead of having carbon have either nitrogen or silicon. Turns out that these nanodiamonds, or these silicon or nitrogen vacancies, are the world's most sensitive material to electric and magnetic fields, which means that if you can actually attach them next to the plasma membrane, they would be the ideal chromophores to sense voltage, because first of all they don't blink, they last forever as diamonds do, and second of all they're super sensitive, several orders of magnitude more sensitive to electric fields than the typical organic chromophores that we use for voltage imaging. No? So maybe through this is work we've done with Jonathan Owen and Dick Erklund at Columbia, derivatizing nanodiamonds, and we're still not there yet, but the idea is to make them stick to the plasma membrane within the region of the Debye length, so that you can actually optically measure these things. Okay, the voltage imaging is one challenge that's ahead of us. That likely requires the effort to many, many groups. It's not like a single lab can fix the problem. Another problem, which is quite fundamental, has to do with the fact that the brain is a two-dimensional structure, but essentially everyone images the brain is a three-dimensional structure. they run images in 2 dimensions. And particularly for laser imaging, laser scanning, you do it by moving the laser back and forth across a plane of focus and then moving the focus down. So this is a major problem, because if you really want to analyze and image complete neural circuits, the more levels, the slower you're going to go. It's a bottleneck. So how to deal with that? So we introduced the use of spatial light modulators, which are essentially liquid crystals. You can change the phase of incoming beam of light, and you can use it to arbitrarily create through constructive interference or destructive interference any pattern of light that you want. You craft the light in 3 dimensions. You build a hologram. You can write words with it with 2 photon lasers. You can draw the pictures of Cajal on top of your brains. And using these SLMs, you can, instead of imaging sequentially, you can image in parallel several neurons, because you split the beam into different beamlets and each of them goes to the target of interest. So this solves parallelizes the imaging. It solves this serial problem of scanning and enables you to, for instance, take a neural circuit like this one, find out what the cells are, build a mask which is uploaded into SLM, and from that point on, there's 100 beamlets, each of them going to every one of these cells, and you simultaneously can image the fluorescence that comes from them using a camera. So this is good to send light in 3 dimensions to many hundreds of thousands of neurons, but there's still a fundamental problem to deal with, and that's the fact that since the time of Leeuwenhoek, microscopes have been designed to image, to focus on a 2 dimension, on a plane. And now we need to be able to actually simultaneously gather the light that's coming from all these different focal points at once. And this is, we have to change the PSF, the basic points per function of the microscope, which looks like this in a typical microscope. This is the focal point, and this is how the PSF looks like. And we've turned these PSFs into these little pencils of PSF, which essentially have a 3-dimensional shape in which you can simultaneously gather photons from all depths at once by introducing a cubic phase mask, which is the diffractive optical element that you can calculate, and you can actually insert it in the emission pathway of the microscope. And when you do that, you can actually achieve what seemed impossible before, which is to simultaneously image things that are out of focus. So this is a typical, this is a mock demonstration where we have a pattern of light that's projected at the focal plane in a conventional microscope, and another pattern that is going up and down in the focus for about 1 millimeter. And in the left case, this is the conventional microscope. The right case is a microscope that has one of these face masks in it, and you can actually see the pattern that's out of focus over a stretch of about 1 millimeter in depth. No? So by changing the PSF to this little type of pencil type PSF, you can start to gather the photons out of these different focal points. We've applied this also in vivo to image the activity of zebrafish, and this is in collaboration with Misha Arens at Genelia. And you can simultaneously gather this calcium imaging of a larval zebrafish, and you can image simultaneously neurons, in this case 50 neurons that are located at different levels of depth, and their fluorescence is gathered simultaneously precisely by aberrating the points per function using this cubic phase mask. Something very quickly, I'm going to tell you about how to turn cells on with light. So there are different ways to do it. One of them is, again, going back to the chemist, and in this case, the inorganic chemist that builds solar cells using the metal ruthenium. Turns out ruthenium can be used to build these very efficient light antennas. In this case, it's a ruthenium bipyridine complex, has great efficiency in gathering photons. You can actually derivatize it through a nitrogen and build a cage neurotransmitters. In this case, it's cage glutamate that binds this ruthenium light antenna, and you can actually release it with light. The metal bond breaks, and within 40 nanoseconds, you've released your molecule of choice in the tissue, and this works with 2-photon excitation. So you can take a bunch of neurons, label them, or bath apply this ruby glutamate, ruthenium bipyridine glutamate, and then go there with your SLM or with your laser and cage on top of the neuron, and you make it fire. And if you move your 2-photon beam just a few microns away, the neuron doesn't fire. So you have single-cell precision in firing neurons one by one using these ruthenium bipyridine complexes. And this is illustrated here. You can actually, again, image a large brain circuit, find out where the cells are, calculate the trajectory, and go and turn them on one by one. And then you can use this to map neuronal circuits. Another approach, instead of optochemistry, is to use optogenetics. In this case, it's C1V1, which is a mixed channel rhodopsin that has a very long time constant, and you can actually transfect it into a neural circuit, into the cortex in this case, and then using special light modulators, you can stimulate the two cells that are literally almost on top of each other with 20 microns distance in C. You can fire both cells together, you can fire one cell, or you can fire another cell, again proving the point that you can have single cell specificity with two photon optical section properties, in this case using optogenetics to turn things on. How to turn things off? So we've done it also with these ruthenium bipyridine complexes, in this case with coupled GABA, the inhibitory transmitter, and we can release it on top of a neuron. It has the right reversal potential. We can use it to map GABAergic currents on cells. Or just to give you an example for what could be the neurology of the future, we've used Ruby GABA to stop epilepsy in a mouse, in this case in collaboration with Steve Rothman at the University of Minnesota. So this is a mouse model of epilepsy where the mouse is undergoing these 4-AP-induced seizures. These are very similar to the ones that are clinically observed in human. We applied to the top of the cortex topically Ruby GABA, which is just water soluble, and the mouse continues with the seizures, and then we turn on an LED and we uncage Ruby GABA, and we stop the seizures for about 2 minutes until the Ruby GABA that's uncaged, the GABA that's uncaged essentially washes it away. So imagine, in particular, these patients that have chronic epilepsy that don't respond to pharmacological therapies, whether in the future maybe you may be able to use this type of light-based therapies to essentially control seizures just with light. So how to play the piano. So let me show you an early example where we're playing the piano with one hand. In this case, we use 2-photon uncaging of glutamate and 2-photon calcium imaging to simultaneously image the activity of 50 neurons, illustrated here from, and by each line is a neuron, and every one of these tick marks is whenever the neuron fires an action potential. And then simultaneously we're going there with a laser and firing 5 neurons at a time, as if we're playing these sort of chords on the circuit, just by pressing 5 keys, one after the other, 5 after the other, marching our way down the circuit. And looks at how interesting these things, this type of experiments could be to decode the transfer function. As we do this, there's some activity floating through the circuit, but when we hit time 200, we fire these 5 neurons here, and suddenly the whole circuit responds, as if we just touch a fiber in the circuit, boom, something has gone off. And then as we continue our march to the circuit, you start getting echoes of that. The circuit essentially is a living tissue responding to our stimulation, our very dumb stimulation, and we go in there blind, so to speak, but it's telling us that it's responding with groups of neurons that are firing together. So this could be a platform to do this, hopefully using spatial light modulators, partly because you can actually make them miniaturize. You can miniaturize the whole microscope design into a little pocket scope, and it's not too far-fetched to think you could actually mount this on top of the skull of a patient and maybe do this type of piano type of experiments on patients that have epilepsy or schizophrenia and try to see if you can actually alter their discharges. Now, all of this is work that was done by a single lab in a little bit amateurish fashion, I should say, and more than anything points us painfully the long road ahead. And every one of these technologies that I told you, for everything that we've done, it's a little step in a long, potentially sort of much more effective type of technology. And that's when we suggested the idea of coordinating and crystallizing a large-scale scientific project, which we call the Brain Activity Map. Together with some colleagues, I was able to convince some nanoscientists and neuroscientists that this wasn't completely crazy, and we wrote proposals that we circulated to different agencies, including the White House Office of Science and Technology. And we made a proposal that neuroscience was right now to carry out a large-scale scientific project, the scale of the human genome, that could have these 3 stated goals: to measure every spike from every neuron in a complete brain circuit of an animal or a human. You could start with small animals like worms. This should be relatively easy, only 300 neurons. Fly, about 100 50,000 neurons, fish, and the adult fish about 1 million neurons, so the mouse, a few million, and eventually learn our technology as we march our way, just like in the human genome, until we can apply it to the human. Goal 2 would be to manipulate the activity of the neurons, so that, as I was telling you earlier, it's not sufficient to just watch. You have to actually get in there with your tools to be able to test causal hypotheses on how these circuits work. And the third goal, which was very nicely explained by Nathan earlier, is to computationally analyze and model this type of circuits, because we really need to develop the computational tools to deal with this emergent level of brain function. To give you a feeling what this type of technology we're thinking about, so not just opticals like the ones I was demonstrating today with calcium and voltage indicators, but electrophysiological approaches that could be very useful for the other speakers in this session, built using perhaps microfabricated chips based on silicon, flexible electronics. I was discussing earlier with Fred Lamy, next generation photonics-based probes, maybe with nanoparticles. And there are synthetic biology approaches. Actually, I just saw Tony Sater walk in that hopefully will tell us about this type of approaches that can be used to reconstruct action potential by sequencing or by using polymerases. And I would point out that these tools are also good for glia. It's not that we leave glia out of the equation. No, these are the same tools can be applied to study neurons and glia, because the glia are very important and relatively still mysterious part of these circuits. How would you develop tools to manipulate the activity of every neuron? I gave you examples of the genetics and cage compounds. You could do this also with nanoparticles. The nanotechnology in the last 10 years have developed a whole slew new types of approaches that haven't been really tapped by the biologists or the neuroscientists in particular. And this could be applied also for the chemical modulation, and of course you could have genetic strategies. And finally, computational approaches need to be developed, not just to deal with these immense amounts of data that are going to be probably larger than what's done at the human genome, but maybe comparable to the types of data that the astronomers are talking about. But more importantly, to create computational models and new math, really, to analyze this data and try to extract what are these ensembles that I was earlier talking about. So just a rough roadmap of how something like this could develop. Imagine that this could be the milestones at 5, 10, and 15 years from now. 5 years from now, how about imaging 50,000 neurons? What could you do with that? You can image the entire C. elegans nervous system during a behavioral relevant period of time, all the mitral cells in the factory bulb, all the retinal ganglion cells in the mouse. The embryo of the zebrafish is actually getting there. What's been done? I mean, the idea would be to measure these things at a kilohertz so you can catch a reaction potential in 3 dimensions. Then with 1 million neurons, you can actually start reconstructing the activity of entire brains of entire animals, like the Drosophila, the entire human retina, cortical area of a mouse. There's a little mammal, the smallest mammal, the Truscan shrew, whose cortex has 1 million neurons per hemisphere. So you could image the activity or measure the activity of 1 million neurons. Maybe you could actually reconstruct the entire cortical activity of a living Truscan, living mammal. And with 15 years, hopefully this technology will be developed enough, and I cannot tell you exactly which one could be the one that could be applied to humans, but the idea would be that it could be applied to awake primates, to human patients, and maybe help in this problem with the brain-computer interfaces. So how can a project like this happen? So we were initiating this project by convincing and inspiring administrators, and it looks like this has happened, and now NIH, NSF, DARPA, and others are both public and private sources of funding are being committed to carry this out. This is going to be a long-term project. It's going to last 15 years. And private foundations have been critical in launching this, in helping to launch this. Particularly the Kavli Foundation was behind us all the way to try to get our message across many of these agencies. And we hope that the industry will actually pick up the slack as the project develops, because besides these great implications for the clinic and implication just to understand ourselves, understand our minds, there is probably a huge area of further economic development, just like what happened with the human genome, that could emerge from the development of this neurotech. You can imagine that just the genomics has come out of the human genome, or you can imagine what could come out and the impact of the technology that would come out from really understanding, being able to measure the activity of the human brain and controlling the activity of the human brain or altering that. Just wanted to finish with a quote from Sidney Brenner, who said that progress in science depends on new techniques, new discoveries, and new ideas, probably in that order, that even though we maybe fool ourselves thinking that the most important thing for a scientist is to test a hypothesis, at the end of the day, you can only see as far as your techniques, and your hypotheses are bound by shackles by your techniques. And I would point out to you that the neuronal doctrine, the reason it's buried in neuroscience as our current dogma, has to do strictly with the fact that the techniques that the anatomists and physiologists have been using for 100 years have been single neuron techniques. Well, no wonder they thought that single neurons are important, because this is the only technique they could use. So I would say, let's develop new techniques. Let's get rid of the neuronal doctrine that served us well for a century. Let's move on and try to tackle this issue of emergent level properties of neural function. And just to finish, I wanted to put a plug for a new institute, or maybe center, that we're trying to build at Columbia, Interdisciplinary Center for Biological Imaging and Neurotechnology, and hope we're encompass about 20 labs in the 3 schools, Arts and Science, Engineering, and the Medical School, and welcome all of you to visit. Thanks a lot. With 2 photon, actually with 3 photon, you can go down about 3 millimeters. And that would get you with the skull open. There's 2 types of humans: skull open humans and the rest of us. Skull open humans, and in case of neurosurgery or human patient, you could just go through the depth of the cortex with stronger lasers than the ones that we have now, and it could be custom made. But you could do that from above. But another very interesting approach is to do it from underneath. So, for instance, there's quite a revolution going on in terms of endovascular sort of manipulation, really, of the human brain. It's not just to image the capillary bed, but there have been new developments in which you can actually insert fibers through the arterial wall into the brain at any particular position that you like. So it is not, and this has been done. I just came from Sweden, and I saw the surgeons do this. And they call it they essentially penetrate the arterial bed. Well, it's relatively non-invasive because it goes through the femoral artery, and then they through a micro guide, a microcapillary, they actually push up a particular probe that in this case it just looks like a needle, but it could be an optical probe or an electrical probe, or it could be a probe with remote sensing capabilities, and you could actually put them in any place of the human brain you want. And the condition in which it's actually clear that it's not doing any damage, and it's preventing hemorrhages because it's plugging the artery as it goes through. No? So maybe another way to think about it is not to try to go from above, but to try to go from underneath. No? And I wouldn't argue that you could image the activity of every neuron in the human brain, but I would say that could be possible to image a relatively dense collection of neurons in any particular interesting area of the human brain in a living patient. So those are examples. Another type of technology that's been developed is there's 2 papers published last year, which they were able to image through an eggshell. And this appears to be impossible, but you can actually put an object, a fluorescent emitter, on the other side of an eggshell, or something similar with the optical properties of an eggshell, and then using a laser and computational optics actually decode the scattering transfer function, so to speak, and figure out and see what's on the other side. So this was done in the University of Colorado and in a group in Amsterdam. So maybe this is today. Maybe 10, 15 years from now, how about imaging through the skull? Then you may be able to apply some of these things through to the rest of us, non-invasively. No? Yes?
So, studies do you go from there?
Yeah, so the data problem is actually really not much of a problem, to our surprise. We organized several workshops, and one of them we invited people from different experts in large-scale data, genomics, astronomers, and particularly Google, Amazon, Facebook, and they look at our calculation and they said, not a problem in terms of the number of petabytes. I mean, this is sort of some back-of-the-envelope calculation with compressed data, compressed being, assuming that's just 1 bit data. 1 million neurons at a kilohertz, that would be about 10 gigabytes per second, so let's say about 36 terabytes per hour. That's not a problem today for these companies, which they have proprietary, they can tell us exactly what is how much data they're handling, but they didn't seem to be worried about the ability of neuroscientists to get this data. But the second part of the question is, well, okay, imagine we solve this data handling problem, and we put that data uploaded in the web or a server somewhere, just like the human genome, and then anyone can mine it. How are you going to mine it? And then my gut feeling is that we actually still need a new type of mathematics, of emergent level neural networks, that there's a lot of people working on computational neuroscience, working in developing these tools, but I still think it's relatively early days, and that we need a lot of theory to help us make sense of that data and know what is the right dimensions that we need to analyze there. No? So I would argue that I would make a pitch that is as necessary as developing the tools is to develop these computational approaches, and that they should be, and that's why we put this third aim to bring in theories and computational types from other fields also to help us with this. Yeah.
A complete neurocircuitry obviously can cross different brain areas in the cortical, right? And that's beyond the optical imaging range for 0 millimeter.
That depends on the animal, yeah. But
right. So do you envision, or how, or should this mapping project interface with other modalities that are better in like connectivities or different big regions of the brain?
Yeah, obviously, I mean, our goal is our pitch is to develop new tools. As this tool building, technique development, this is in the sense that it gets to the point where, as I was making the argument, you only see as far as your tools, and it's about time that we leave behind the single neuron approaches. And I would make the argument that these tools cannot be developed by individual labs. This is the time in science where the science grows to be a mature science, and just like astronomers have observatories and chemists have and physicists have national laboratories with particle accelerators, maybe neuroscience should have large-scale scientific project where people can actually get at these problems and make systematic effort to solve them. No? But okay, but you're right about this large-scale connectivity. For small animals, I think it's possible to image every neuron in the entire brain of the animal. And this is starting to be done, for instance, in the zebrafish, in the larva, although at very slow time resolution, about 0.8 hertz. But it's getting there. I mean, if maybe in 5 years you can do that at a kilohertz in a zebrafish, then you have every neuron. Then you cannot tell me there's more stuff going on. Say, no, that's it. It's not like there's another nervous system somewhere. That's it. That behavior is based on these neurons, and there's nothing more, no more magic. So that's the beauty of a complete reconstruction, that you said the buck stops here. But what happens in the human? So in that case, I don't know, a reasonable thing would be to analyze particular areas that are of critical importance to particular parts of neuroscience. I don't know, maybe epileptic reconstruction of the temporal lobe or something like that. But of course, not to do this in the vacuum. I mean, we expect that this tool will be used by neuroscientists that would be embedded in the neuroscience of their times, and they will be knowledgeable about all the connectivity and all the biophysics and cellular properties of the system, so that they can actually use that to their advantage. Yes?
I think I want to argue with your last slide where you quoted grammar. So I'm coming from a different perspective than talking neuroscience. But I was just reading some things about the history of the Niels Bohr-Einstein debate on quantum entanglement. Einstein proposed a theory back in the 30s. It wasn't until the 1980s that developed a technique that could be verified. But the theory led by far and away in this kind of experiments with techniques. And I do feel like when you put up these numbers, they say, well, in the worst case, we get 30 petabytes of imaging data from mouse brain or whatever. But even if you could have the catalog on the web, the whole thing, it just seems to me that without really good theory leading the way,
Yeah.
it's completely just garbage in, garbage out.
Well,
And we
yeah.
really do need to commensurate with new techniques, a new way of thinking about
Okay.
models of behavior in a complex system like the brain.
Yeah, okay, so I wouldn't say it's this, I wouldn't characterize this as garbage, but more or less as the, this would be the permanent.
Garbage theory and garbage out.
Yeah, I mean, I agree that we need better theories. I just argue that, and there could be cases in science, and I won't argue with that, where theory has led the way in the absence of any experimental evidence. Now, this applies, in my mind, more to physics and particular branches of physics and to chemistry than to biology. I think in biology, I see biology more as discoverers. We're entering an unexplored territory. It's very difficult to calculate how things are going to be from first principles. So there's been a long track record of theories being wrong in biology, and very good theories, and being completely wrong. Because these are systems that could be very nonlinear, where also the logic is not really one that we have access to, because it's probably an evolutionary logic, something that happened millions of years ago, and has to do with behavior that we may not exist now. So it's a little, I would say, risky to be too theory heavy in this particular case. No, but I'm just pointing out that from a frank analysis of the history of neuroscience, and if you agree with me that the neural doctrine is the core of what is taught today, I would argue that this is strictly a technical limitation that had to do with these Golgi methods that only label single cells, and electrorecording that record from individual neurons. And that I presume that we have, just in this unexplored continent, we have all these other areas that no one has ever been there. No? But yeah, I mean, I couldn't agree more that you have to have theories that go hand in hand with these new methods. No? Yeah.
We should just wrap up. Thank you, Rafael.